Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis of classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two kingdom system of classification with Plantae and Animalia kingdom was developed that include all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green alga and non-photosynthetic fungi organism. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationships, etc. Classification system for the living organism have hence undergone several changes over the time, though plant and animal kingdom have been a constant under all different systems. The understanding of what groups organisms to included under these kingdoms have been changing. The number and the nature of other kingdom have also been understood differently by different scientists over the time. Table 2.1 Characteristic of five kingdom. Characters, cell type, cell wall, nuclear membrane, body organization, and mode of nutrition. Five kingdom Monera, Protista, Fungi, Plantae, and Animalia. Cell type in Monera prokaryote and rest of them eukaryote. Cell wall in Monera non-cellulosic polysaccharide plus amino acid. In Protista present in some. In fungi present with chitin. In Plantae present with cellulose. Animalia absent. Nuclear membrane in Monera absent. Rest of them present. Body organization Monera cellular, Protista cellular, fungi multicellular loose tissue, Plantae tissue and organ, Animalia tissue organ and organ system. Mode of nutrition in Monera heterotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic and parasitic. In Protista heterotrophic, photosynthetic and heterotrophic. In fungi heterotrophic, saprophytic and parasitic. In Plantae heterotrophic, photosynthetic. In Animalia heterotrophic, holozoic and saprophytic, etc. R.H. Whitaker in 1969 proposed a five kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 give a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain and thereby a six-kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five kingdom classification to understand the issues and considerations that influence the classification system. Earlier classification systems include bacteria, blue-green alga, fungi, mosses, ferns, gymnosperm, and the angiosperm under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their walls. These placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green alga cyanobacteria with other group of which were eukaryotic. It also grouped together the unicellular organism and the multicellular one. Say, for example, Chlamydomonas and Spirogyra were placed together under alga. The classification did not differentiate between the heterotrophic group, fungi, and the heterotrophic green plant, though they had shown a characteristic difference in their wall compositions. The fungi had chitin in their wall, while the green plant had a cellulosic wall. When such characteristics were considered, The fungi were placed in the separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which is earlier classification were placed in a different kingdom. This happened because the criteria for classification change. These kind of changes will take place in future too, depending on the improvement in our understanding and characteristic and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristic of kingdom Monera, Protista, fungi of the Whitaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapters 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole members of kingdom Monera. They are the most abundant microorganisms. Bacteria occurs almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, deserts, snow, and deep ocean where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, plural cocci; the rod-shaped bacillus, plural bacilli; the comma-shaped Vibrium, plural Vibrio; and the spiral, plural Spirilla. Figure 2.1. Though the bacterial structure is very simple, they are very complex in behavior compared to many other organisms. Bacteria as a group show most extensive metabolic diversity. Some of the bacteria are heterotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic heterotrophic or chemosynthetic heterotrophic. The vast majority of bacteria are heterotrophic, that is, they depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitat, such as extreme salty area, halophils, hot springs, thermoacidophils, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green alga, have chlorophyll A similar to green plant and the photosynthetic heterotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater, marine or terrestrial alga. The colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example: Nostoc and Anabaena. Chemosynthetic heterotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature in majority of important decomposers. Many of them have a significant impact on human affairs. They are useful or helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogenic, causing damage to human beings, crops, and farm animals and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figure 2.3. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known as and can survive without oxygen. Many mycoplasma are pathogenic to animals and plants. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protistian to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primitively aquatic. This kingdom form a link with other dealing with plants, animals, and fungi. Being eukaryotes, the protistian cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella and cilia. Protista reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. These group include diatoms and golden algae desmids. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water current plankton. Most of them are photosynthetic. In diatoms, the cell wall form two thin overlapping shells, which fit together as a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producer in the oceans. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigment present in their cells. The cell wall has stiff cellulosic plate on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellates, example guanulex, undergoes such rapid multiplication that they make the sea appear red tides. Toxins released by such large numbers may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of euglenoid are identical to those present in higher plants. Example, euglena. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies, bearing spores at the tips. The spore possess true walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predator or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, false feet, as in amoeba. Marine forms have silica shells in their surface. Some of them, such as entamoeba, are parasitic, flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic form cause diseases like sleeping sickness, for example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that open to the outside of the cell surface. The coordinated movement of the rows of cilia cause the water laden with food to be steered into the gullet. Example, paramecium. Sporozoans. These include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite which causes malaria, a disease which has a staggering effect on human population. 2.3 Kingdom Fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and the toadstools are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make beer and bread. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occurs in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from bad due to the malarial, bacterial, and fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long cylinder thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called cenocytic hyphae. Others have separate or cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence are called saprophytes. Those that depend on living plant and animal are called parasite. They can also live as symbiotics in association with alga as lichen and with the roots of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore called conidia or sporangiospore or zoospore, and sexual reproduction is by oospore, ascospore, and basidiospore. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. Step number 1, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Number 2, fusion of two nuclei called karyogamy. Number 3, meiosis in zygote result in the haploid spore. When a fungus reproduces sexually, two haploid hyphae of compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in the diploid cells 2N. However, in other fungi like Ascomycetes and Basidiomycetes, an intervening dikaryotic stage N plus N, that is 2 nuclei per cell, occurs. Such a condition is called a dikaryon, and the phase is called dikaryophage of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to the formation of haploid spores. The morphology of the mycelium, mode of spore formation, and fruiting body form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospore motile or by aplanospore non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by the fusion of two gametes. These gametes are similar in morphology, that is isogamous, or dissimilar, that is anisogamous or oogamous. Some common examples are mucor, rhizopus, the bread mold mentioned earlier, and the albugo, the parasitic fungi on mustard. 2.3.2 Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast Saccharomyces. They are saprophytic, decomposers, parasitic, or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spore are conidia produced exogenously on the special mycelium called conidiophore. Conidia on germination produce mycelium. Sexual spore are called ascospore which are produced endogenously in sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some example of Aspergillus, claviceps and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacies. 2.3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, bracket fungi or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasite. Example rust and smut. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative and somatic cells of different strains or genotype. The resultant structure is dikaryotic, which immediately which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium producing four basidiospores. The basidiospores are exogenously produced on the basidium plural basidia. The basidia are arranged in fruiting bodies called basidiocarp. Some common members are agaricus mushroom, Eustiligo smut, and Puccinia rust fungus. 2.3.4 Deuteromycetes, commonly known as imperfect fungi, because only the asexual or vegetative phase of these fungi are known. When the sexual form of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given the name and placed under Deuteromycetes, and the sexual stage another that is placed under another class. Later, when the linkage were established, the fungus were correctly identified and moved out of Deuteromycetes. Once perfect sexual stage of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduced by a sexual spore known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposer of litter and help in mineral cycling. Some examples are Alternalia, Coletotrichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophs, such as insectivorous plant or parasite. Bladderwort and Venus flytrap are examples of insectivorous plant, and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae include algae, bryophytes, pteridophytes, gymnosperm, and angiosperms. Life cycle of plant has two distinct phases, the diploid sporophytic and the haploid gametophytic, that alternate with each other. The length of the haploid and diploid phases and whether those phases are free living or dependent on other vary among different groups in plants. This phenomenon is called alternation of generation. You will study further details in this kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell wall. They directly or indirectly depends on plant for food. They digest their food in an internal cavity and store food reserve as glycogen and fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female, followed by embryological development. Salient features of various phyla are described in chapter 4. 2.6 Viruses, viroids, prions, and lichens. In the five kingdom classification of Whitaker, there is no mention of lichens and some acellular organisms like viruses, viroids, and prions. They are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effect viruses can have on us. Even if we do not associate it with your condition, viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the virus and non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes as casual organisms of the mosaic disease of tobacco. Figure 2.6a. These were found to be smaller than bacteria because they pass through bacterial proof filter. M.W. Bajornik in 1898 demonstrated that the extract of the infected plants of tobacco would cause infection in healthy plants and named the new pathogen virus and called the fluid as Contagium fivum fluidum, infectious living fluid. M.W. Stanley W.M. Stanley 1935 showed that viruses could be crystallized and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No virus contain both RNA and DNA. A virus is a nucleoprotein and a genetic material is infectious. In general, viruses that infect plant have single stranded RNA and viruses that infect animals have either single or double stranded RNA or double stranded DNA. Bacterial virus or bacteriophages virus that infect the bacteria are usually double-stranded DNA virus. The protein coat called capsid, made up of small subunit called capsomere, protect the nucleic acid. These capsomere are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptom can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing, and stunted growth. Viroid. In 1971, Theo Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat is found in viruses. Hence the name viroid. The RNA of viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to virus. These agents were called prions. The most notable disease caused by prion are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant CR-Jacob disease (CJD) in humans. Lichen. Lichen are symbiotic associations, that is mutualism, useful association between algae and fungi. The algal component is known as phycobiont and the fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichen are a very good pollution indicator. They do not grow in polluted areas. Summary. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, that is, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationship. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophyte, dinoflagellates, euglenoids, slime molds, and protozoans. Protists have defined nucleus and other membrane-bound organelle. They reproduce both asexually and sexually. Members of kingdom fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are the four classes under this kingdom. The Plantae include or eukaryotic chlorophyll-containing organisms. Algae, bryophyte, pteridophyte, gymnosperm, and angiosperm are included in this group. The life cycle of plants exhibit alternation of generation, gametophytic and sporophytic generation. The heterotrophic eukaryotic multicellular organism lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these animals or organisms is holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like viruses and viroids as well as lichens are not included in the kingdom 5 kingdom system of classification. Thank you. Thank you everyone.